Good afternoon, everyone. This is Greenson Sharma. Today I'm going to talk about the recombinant DNA technology. This is the technology which includes the process of a combination of a molecule of DNA from two different species which are inserted into the host cell to produce a combination that have some value to the medicine as well as the technology. And the fundamental goal of any researcher is to isolate and to characterize the genes. And in this video, first we will be talking about the cloning. We can call it either DNA or the gene cloning. Cloning is just the process of production of the same copy. Here you can see in the figure, this is the iPhone 11. You have a mobile phone and you are making the phone of same structure again and again. That is generally known as cloning. A particular model of a smartphone having the multiple copies is known as clone. Here the cloning is for DNA or gene, creating the multiple of same copies of DNA or gene using the bacteria as the host organism. Generally we use bacteria as the host organism. And let us go into the requirement portion. Generally the requirement is a gene of interest is obvious, which can be cloned by the process of the gene of interest can be amplified by the process of PCR. And for example, the human insulin, generally gene of interest is kept to produce the more amount of gene of interest in this case. And for the vector, vector is the transport system of gene to the host cell. And for the amplification process, the E. coli is generally needed. It's generally the insertion of a gene with a vector into the host cell. We can also call it E. coli. And after that, the extraction is generally the process of extracting the required gene of interest, amplified gene of interest from the host cell. Here we can see the figure also. This is the vector, this is the DNA. Here we can we convert this fragment of DNA and vector in the recombinant DNA, and we insert that into the bacteria, and it's a multiply, and finally we can get the bacterial colonies. Gene of interest can be amplified by using the polymer chain reaction. It is the thermocycle. Here multiple copies of target DNA can be made by using the polymer chain reaction. Here you can see the PCR machine, and we are going to follow the PCR cycle in this way. First of all, the requirements are the target DNA, which need to be amplified, the primers, the nucleotides is obvious, and the polymerase enzyme, and they are generally mixed into the PCR tube, and they are kept in a PCR machine for various cycle. And in the PCR machine, here you can see that it's the technique that perform the thermocycle by heating and cooling down the temperature in generally 3 steps: the DNA denaturation, annealing, and the extension. Here you can see that in the cycle 1, the denaturation is being done at 95 degrees Celsius, and the annealing at 55, and extension at 72 degrees Celsius. First of all, due to the action of polymerase and the primer and the nucleotide in the 3 cycle, the first DNA can be formed like in this figure. You can see, first of all, the primer will go at the respected place, and due to the action of polymerase and the free nucleotide, you can see the formation of the double stranded DNA from the denatured DNA. Here we can see the 2 DNA was formed. And in this method, after each cycle, the number of PCR sequence will be doubled by 2n number of times, just like in the second cycle. It is the same process. That is, first the action of primer, the polymerase, and the a nucleotide in various three steps: the denaturation, annealing, and extension. Here we can see that the 4 DNA was formed, 4 DNA strand was formed. Also in the cycle 3, the process is the same, just like denaturation, annealing, and the extension. And you can see the same cycle. It is generally the process of a DNA amplifying the specific sequence of the gene. And other than the real-time PCR, the time can be set in PCR, and at the last of thermal cycle, the amount of amplified DNA at the sample can be measured. But during the cycle, the amount of amplified DNA cannot be measured, and this deficiency is fulfilled by the real-time PCR. Here we can see that with increasing the number of cycle, the copied number of target DNA is also increasing, and at the last 30th cycle, we can get about 1 billion copies of DNA from the single in 4 hours from the single stranded of DNA. Let's discuss about the vector. To be a vector, it should have the property to carry a segment of target DNA. Generally, the most common vector is the bacterial plasmid vector, and it is the circular double-stranded DNA which is capable of self-replication, as it has the origin of replication. This bacterial plasmid vector has the origin of replication, and also it has the multiple cloning site, and it also have the selective target marker. And there are various type of vector. Here let us discuss about the cloning vector and the expression vector. For the cloning vector, it is the vehicle of a gene delivery system. For example, we can call it that plasmid vector, and it is used generally for the cloning purpose only, aka for the development of multiple copies of the same gene. In the expression vector, here we can see the expression vector, and these vectors are used for expression of a gene inside a host cell. And this vector, along with the origin along with the origin of replication, multiple cloning site, and a selective marker, this vector requires the promoter, transcription stop and start site, and the tag sequence also. And for the cloning vector, we can also known as, for example, the plasmid vector. These are effective in nature, and the replication is too fast in the plasmid vector. We can get a lot of plasmid vector in a few amounts of time. And the disadvantage of this plasmid vector is the limited length carrying capacity of the plasmid vector, is not all of the host cell can get the same type of vector. Each host cell has the capacity to acquire the specific length of DNA only, and plasmid vector can carry only limited length of DNA, as the carrying capacity is too limited. And if we increase the length, then the plasmid cannot carry the DNA, as the plasmid vector will disintegrated or unstabilized. So for the larger DNA, we can use the BAC, that's the bacterial artificial chromosome, or the YAC, that is the yeast artificial chromosome. Let us discuss about the bacterial transformation. It is the process of gene transfer by which the bacteria will uptake some of the foreign DNA molecules from its environment, just like we can see from the environment. And bacteria having such capacity to uptake the free DNA from the environment is known as the competent cell. Here in this method also I have used the competent cell. The basic principle of the bacterial transformation is the plasmid. Here we can see that it contains the antibiotic resistant gene, and we also we insert that into the competent cell colony. And if this colony can grow in the antibiotic medium, is the plasmid which contain the resistant gene, and we can see different colonies So here in this method, this is the method that I have done in my lab. Here I have used the culture media of agar plus antibiotic, and I use the competent cell with along with the vector, and I vertex and spin down the solution, and I put into the overnight incubation. Here I use the two different type of vector, a pimir glow vector and PMRI vector. And after the following day, I take the 4 ml of liquid broth plus the ampicillin solution, and then I take the single colony and insert it with the tip, and I did the colony formation process of this competent cell. In the following day, I did the plasmid DNA purification process. Here we can see the gel electrophoresis and the result of gel electrophoresis by the PMRI and PMRI glow vector. Here for the harvested cell, I centrifuge and I discard the supernatant, and I use the resuspension solution, and after that I use the lysis buffer, and along with that I use the neutralization buffer, and I wash that solution with the washing buffer, and I make the solution of recovered plasmid in distilled water. And after this plasmid DNA purification process, I use the gel electrophoresis, and the requirements are the vectors in the agar gel. Here 1% of gel is generally used for the plasmid, whereas for the DNA molecule 2% of gel is generally used. And also for the generation of more sample, PCR was done, and these are the contents of reverse primer, forward primer, DNA and nucleotide. Here the annealing temperature was made 65, and it is generally used for 35 cycle. Here we can see all the band that I get in the gel electrophoresis. Here we can check the PCR, the reliability of PCR with the gel electrophoresis. And after the reliability that of the PCR, I use the DNA isolation method, and which can be done by the same process of gel electrophoresis. Here the method is used is that I cut the slice and I put the gel lysis BNL buffer, and I centrifuge, and again with the centrifuge with the washing buffer, and I centrifuge for to dry the membrane, and after that I use the elution. Instead of elution buffer, I generally use the distilled water of about 30 microliter, and I get the pure DNA fragment. And after that also I did the gel electrophoresis, in which I get the band. Here we can see that. Let us talk about the restriction enzyme. This enzyme are used to cut the DNA sequence. Here we can see that these are the restriction enzyme, just like in III, EcoRB and PSTL. These are the restriction enzyme, and these are generally used to cut the DNA. Here we can see that the cutting of DNA. Here you can see that the 5' protruding in and 3' protruding in. Here you can see the blunting. Actually, by the action of there is generally two type of cutting actually of the sequence, that is the blunt end or the protruding end, or that's we also known as the sticky end. And generally the restriction enzyme break the phosphodiesterase bond which are present in a different nucleotide sequence, forming the blunt end, the blunt or the sticky ends. In this enzyme acts on the three function, that is the recognition of the sequence to cleave, and the second function is to cleave the cleavage of the sequence, and third one is the modification of the sequence. Here we can see the property of the restriction enzyme, that is the specificity. They are very much specific for the cleavage of the DNA, and they are also attached to the specific recognition sequence of the DNA. And the another property of the restriction enzyme is it generally cleave only double-stranded DNA, and they generally produce the different fragments, just like the sticky or the blunt ends. And the sticky ends are very easy for ligation. After the restriction enzyme, we have to do the ligation for the recombinant DNA to form the recombinant DNA, and for that the stickies can be like easily in for the blood things. They are difficult for the ligation. This is the process that I use in my lab for the restriction enzyme activity with DNA. For the DNA in the vector, and for the requirement I use the 2 microliter of DNA in vector, and the BAM I and the HIND I are the restriction enzyme, and this is the NAB buffer that generally for this the buffer for this enzyme. And I also use some distilled water, and the total sample was the 100 microliter of the sample, and I did the overnight incubation. And the following day for the purification process I used the BNL buffer, and I also used the washing buffer of about 750 microliter. And after that I used the column drying method for about 18,000 rpm per 3 minutes, and I make the recovered plasmid DNA in the recovered plasmid factor in the DNA with in distilled water at about 30 microliter. And after that I have to check whether the restriction enzyme I had put in the DNA vector was good or not. That's why I did the gel electrophoresis process. Here in the PMIR vector I use the 1% agarogel, and in DNA I use the 2% agarogel, and here you can see the band. And so I can say that the restriction enzyme work good in the both vector and the agarogel. Let us discuss about the ligation. Ligase is the enzyme that link the two DNA sequence together, and the process is called ligation. When there is missing of the nucleotide in the DNA, the gap is formed, whereas when there is the breakage of the phosphodiester bond, there is the formation of the nick between the nucleotide, and DNA ligase helps to seal the nick between the nucleotide, but not the gap. Here you can see the difference between the nick and the gap. In cloning, after the action of the restriction enzyme to produce the recombinant DNA, that it to seal the nick, the DNA ligase is used. And generally, T4 ligase is widely used than the E. coli DNA ligase. Because T4 ligase can link both the sticky and the blunt end, whereas the DNA ligase, whereas the DNA E. coli ligase can link only the sticky end, but it cannot join the blunt end. That's why. And also the T4 DNA ligase use the ATP is the energy source, whereas the E. coli DNA ligase use the NAD plus is the energy source. That's the general difference between the DNA ligase and E. coli DNA ligase and T4 DNA ligase. And here is the requirement that I use for the ligation. The requirements are the obvious the DNA in the vector which needs to be ligase, in 10x buffer in T4 ligase, and for the total I use the 20 microliter, and I do the ligation over the overnight in at 16 degrees Celsius. And after here you can see the basic principle of ligation, that this is the vector, this is the gene of interest, and after the restriction enzyme and the ligation we can see here is the recombinant DNA molecules, and after the formation of the recombinant DNA molecules, we can put the DNA inside the bacteria, and here is the multiplication of the recombinant DNA molecule, and after that there is a division, and different colonies can be formed. Here also I have formed the 5 colonies, and for this the bacteria transformation process, the ligated mixture I take about 10 microliter and the competent cell about 20 microliter, and I incubate that in for ice incubation at 20 degree, and for 1 minute I use the 42 degrees Celsius of heat, and again after that 20-minute ice incubation, and after that the 20 microliter broth, and I put that into the agar plate overnight, and finally I get the 5 colonies, and after that I have to purify that colony, and for that I have the harvested cell are put into the resuscitation buffer, and then again with the lysis buffer and neutralization buffer, and again with the washing buffer, and I centrifuge that at 13,000 rpm for 1 minute, and after that finally I made about 30 microliter of recovered plasmid DNA with distilled water, and to check this I use the gel electrophoresis method, and these are the requirement, and I get the band. Here we can see the band for the 4 colonies, the ligated vector of DNA in about 1.5% agar gel, and after that the ligation I use the restriction enzyme so that I could find the if this vector in DNA of recombinant DNA I generally use to cut this portion, and I want to this is separated by the gel electrophoresis. That's why I have used the restriction enzyme, and for that I use the ligated vector in DNA of about 4 microliter, and the enzyme, the restriction enzyme of the respected microliter, and I use the buffer for this enzyme, and also the distilled water was added, and I incubate about 2 hours and 30 minutes incubation at 37 degrees Celsius, and also the loading buffer was added, and I did the gel electrophoresis. But here we can say I can get the band for the vector, but I could not get the band for the DNA. Here we can see here, and I'm still trying to do. I'm also in this week I'm just repeating the same process, and hopefully I'll get the band this week. For the application of recombinant DNA technology, first of all, it is generally applied in the field of molecular biology. The gene mapping is one of the technique in the molecular biology where you can use the recombinant DNA technology. Gene mapping will tell us the exact location of gene in chromosome. Also, in the genetic disorder, we can apply the real recombinant DNA technology method for the identification of the genetic disorder. Recombinant DNA technology can be used, and by designing the DNA probes, the genetic disorder of the body can be identified. Also, for the production of the monoclonal antibody, we can use the recombinant DNA technology where the hybrid cell of 2 different cells are fused, and they start to form the different monoclonal antibody which will bind to the specific antigen, and that's how we can form the monoclonal antibody. And the fourth one is the gene therapy. As you know that the faulty gene or the DNA in our body can be replaced with a better copy of the gene, which also helps to for the mitigation of the various disease. Also, for the production of the vaccine, we can use the recombinant DNA technology, and for the pharma product like the recombinant insulin was made by using the recombinant DNA technology.